Hello friends, welcome to my course on Microbial Biotechnology. Today we will be discussing module 4 where the first lecture is on genome structure, transcription, genetic code and translation. We will also of course start with the discussion on the structure of DNA and RNA and also replication, which is an important part of this lecture. So, let us first once again revisit the concept of genome. You already know that a cell's complete set of DNA is called as its genome. In prokaryotes, the genome is a single circular double-stranded DNA, and this is located in a region which is known as the nucleoid, because prokaryotes do not have a well-defined nucleus. Some prokaryotes also contain a small non-essential DNA loop called as plasmid, but it is not exactly non-essential. It gives the bacteria a lot of advantage in coping to environmental stress like antibiotic resistance, etc., which we have discussed in some of the earlier lectures. And this also helps in metabolizing certain uncommon or toxic substances. Now, let us start looking into the structure of DNA. So, some of the simple molecules like 2-deoxyribose sugar, they combine with nitrogenous bases like adenine, thymine, cytosine and guanine through a glycosidic bond to form a compound which we call as a nucleoside. The 2'-deoxyribose sugar that takes part in this reaction lacks a hydroxyl group at the 2' carbon, and thereby distinguishing it from RNA's ribose sugar, which is a hydroxyl group at that position. When one or more phosphate groups are added to these nucleoside via phosphoester bonds, it becomes what we call a nucleotide. These nucleotides are the building blocks of DNA. So, if we want to know the structure of DNA, it is very much essential to understand what is a nucleoside and what is a nucleotide. So, these nucleotides will further polymerize through phosphodiester bonds to form polymeric strands which have directionality, for example, 3' to 5' or 5' to 3'. DNA consists of two such complementary strands, nucleotide strands, which are coiled into a double helix, with each strand having a backbone made of alternating 2' deoxyribose sugars and phosphate groups. So, this is the simplest way in which we can understand the structure of a DNA molecule, with simple molecules like deoxyribose sugar joining with nitrogenous bases to forming a nucleoside, which again in turn add on phosphate groups to form nucleotides, and these nucleotides then polymerizes to form single strands, and they have complementarity in the DNA double structure where they will be existing in a double helix. So, these nucleotides are linked in a polynucleotide chain, as we have just mentioned, by phosphodiester bonds, and these form between the 3' hydroxyl group of one nucleotide sugar and the phosphate group of another's 5' hydroxyl group. And these linkage creates the sugar phosphate backbone, which is a defining feature of the DNA double helix. There you can see here the sugar residues, and you can see here the sugar phosphate backbone with the phosphate groups over here, and then you see the thymine, adenine, guanine and cytosine over here. Now, let us look into one end of this chain. So, you can see here a 5' level, and alternatively in the other strand we can see a 3' level, and this is one polymer and this is another polymer of the nucleotide polynucleotide chains. So, one end has a 5' free 5' phosphate or hydroxyl group, while the other end has a free 3' phosphate or hydroxyl group. These are referred to as the 3' and 5' groups. This directionality is very, very crucial for DNA replication and transcription. So, these DNA bases, these are flat and then heterocyclic structures made of carbon and nitrogen, and they are divided into two categories. The first one are the purines, where you have the two members adenine and guanine, and they have two rings, as you can see over here, two rings each. Then you have the purines, pyrimidines, cytosine and thymine, which have only one single ring each. And now adenine will pair up with thymine and guanine will pair up with cytosine, and this pairing is stabilized by hydrogen bonds, as you can see here by these dotted lines over here. And from the diagram itself, you can know that thymine and adenine forms two hydrogen bonds, while guanine and cytosine will be forming 3 hydrogen bonds. So, in a double helix, in brief terms, a base pair via hydrogen bonds, adenine pairs with thymine by 2 hydrogen bonds, and guanine pairs with cytosine by 3 hydrogen bonds. The two DNA strands are antiparallel, running in opposite directions, one 5' to 3' and the other 3' to 5', with base pairing occurring along the strands entire length. And these type of AT-GC pairing is what we call as complementary base pairing. And so, the two opposite strands are complementary to one another, only then a DNA double helix can be stable. Most prokaryotic cells contain a single circular chromosome located in the cytoplasm's nucleate region, is already known to you, and E. coli genome has about 4.6 million base pairs. This DNA undergoes supercoiling, which can be underwound or overwound. Proteins and enzymes such as DNA gyrase help maintain and regulate the supercoiled structure, which is crucial for DNA replication, transcription, and fitting the DNA within the small cell. Now, let us look into the structure of RNA or ribonucleic acid, which is an essential nucleic acid found in all cells. While RNA is made from nucleotides like DNA through a process called as transcription, it has several key differences. RNA has a ribose sugar backbone, whereas DNA contains the deoxyribose. Then base composition, also certain differences are there. RNA uses uracil instead of thymine, resulting in base composition of adenine, cytosine, guanine and uracil. RNA exists in single stranded form typically, allowing it to fold and form short double helical structures or segments with the complementary regions. And RNA allows a broader range of base pairing. Uracil pairs with adenine instead of thymine as in the case of DNA. Now, let us look into one of the most important fundamental process, replication, which is basically the doubling of a DNA molecule. So, DNA replication in bacteria like E. coli occurs on its single circular chromosome, which is about 4.6 million base pairs long, as already mentioned. The replication will begin at a single origin of replication called oric, and it will proceed bidirectionally with two replication forks moving in opposite directions. The process is highly efficient, with the DNA polymerase adding around 1000 nucleotides per second, which is very fast, and the entire genome is typically replicated in about 40 to 45 minutes, depending on the strain and the environment in which it exists. The process is highly accurate with a low error rate due to proofreading mechanisms that ensures the fidelity of the DNA sequences. So, you can see here one DNA molecule of a bacterium, and it has a origin. The point in the DNA molecule, double stranded molecule, from where the replication starts, and you can see the replication is bidirectional. It is proceeding in both the directions starting from the origin. So, replications begins at the origin of replication, where the DNA unwinds and is duplicated at the replication fork. In circular chromosomes, the replication process creates a structure resembling the Greek letter theta. So, this structure, as you can see, looks like the Greek letter theta. That is why it is also known as theta replication. As replication will proceed, the two forks move outward, copying the chromosome until they converge on the opposite sides, resulting in two separate DNA molecules or chromosomes. What are the components of the replication machinery which helps in converting a single chromosome or DNA molecule into two DNA molecules or two chromosomes? So, some of the key components are proteins like helicases. They unwind DNA ahead of the replication fork. So, here is the replication fork, as you can see over here, and the helicase is unwinding the DNA ahead of this replication fork. So, since this is trying to open up DNA, it is an energy consuming process. That energy is supplied by ATP. Then there are certain proteins known as single stranded DNA binding proteins or SSBs. So, since these are the bases as depicted in this diagram, once the helicase opens these DNA double strand, because of complementarity, they will again form a double stranded structure. To prevent that, the single strand binding proteins will stabilize the single stranded DNA and keeps these strand open. So, basically they prevent separated strands from re-annealing. Then we have certain enzymes known as topoisomerases. This relieve tension from unwinding because of supercoiling. The DNA molecule is very difficult to open, and since we are opening it up, tension is getting built up. So, to relieve that tension, the topoisomerase will be unwinding the DNA molecule. DNA gyrase is a key enzyme in E. coli in this case. Then we have other molecules like DNA polymerases who do the polymerization work. So, here they add nucleotide in the 5' to 3' direction, which requires a 3' OH group and uses nucleoside triphosphates like DATP, DGTP, DTTP and DCTP as building blocks and the energy source. In E. coli, DNA polymerase III is the primary enzyme responsible for DNA synthesis, while the DNA polymerase I plays a key role in removing RNA primers and filling in gaps. So, these are some of the important components of the DNA replication machinery. Other important ones are the primase, and then we also need a RNA primer which is around 5 to 10 nucleotides long. A DNA polymerase cannot act on DNA template on its own. It requires a RNA primer from where it will start the addition of the nucleotides one by one. So, this RNA primer is synthesized by this enzyme called primase. So, primase synthesizes a short RNA primer which is about 10 nucleotides, which is complementary to the DNA portion to which it binds, enabling DNA polymerase III to begin synthesis. Due to DNA's antiparallel nature, replication differs for the two strands. The leading strand is synthesized continuously in the 5' to 3' direction towards the replication fork, allowing smooth uninterrupted synthesis. The lagging strand is synthesized discontinuously in short segments, which we call as Okazaki fragments, each initiated by an RNA primer. These fragments are made in the 5' to 3' direction, but apparently away from the replication fork. The fragments are later joined by DNA ligase, which is therefore another important component of this replication machinery after the RNA primers are removed. So, we can see that the DNA replication is taking place in two opposite directions. In the leading strand, it is happening towards the fork, and in the lagging strand, where small fragments of DNA are replicated and later on joined by DNA ligase, the replication is in the direction which is away from this replication fork. At the replication fork, DNA's antiparallel structure presents a challenge as the two template strands run in opposite directions. The leading strand is synthesized continuously toward the fork, complementing the 3' to 5' template. The lagging strand synthesized in short okazaki fragments requires a separate primer for each fragment and is made away from the fork in the 3' to 5' direction, while the leading strand runs in the 5' to 3' direction. However, the revised model of DNA replication is not that as simple as this. There is some kind of a adjustment for the steric hindrance problem over here. So, in the lagging strand, there will be a loop formation and there is a reason for these which we will try to explain over here. But you need to remember is that the leading strand replicates towards the fork center, and in the lagging strand there is some kind of looping and not as straight as these. However, the okazaki fragments and other things will be all present in this model as well. So, in E. coli DNA replication, there is a overall, not only in E. coli, beta clamp which will ensure high processivity by holding the DNA polymerase tree onto the DNA template, allowing it to synthesize long stretches of DNA efficiently. The looping model helps overcome the challenge of synthesizing the lagging strand which runs in the opposite direction to the replication fork. By looping the lagging strand template, polymerase can synthesize okazaki fragments in the correct 5' to 3' direction. The beta clamp and looping mechanism work together to coordinate the continuous synthesis of the leading strand and the discontinuous synthesis of the lagging strand, ensuring efficient and simultaneous replication of both the strands. So, if we want to look into the DNA replication in very brief, there is DNA unwinding at the origin of replication, then there is a helicase action which separates the strands, creating replication forks in both directions. Then the strands are stabilized by single strand binding proteins to prevent the strands from reforming a double helix. Then the tension is released by topoisomerases ahead of the fork. Then there is primer synthesis by primase, and this primer is RNA primers. So, we have to remember that the aim of replication is copying DNA to DNA, but that starts with first making a small RNA primer, not a DNA primer. We use DNA primers in polymerase chain reactions in silico methods. So, then there is DNA synthesis where the DNA pol tree adds nucleotides to the end of the primers, elongating the leading and lagging strands. Then the primers has to be finally removed from this DNA molecule, otherwise there will be errors. So, there are exonucleases which removes this primer. Then there is gap filling, DNA polymerase 1 fills the gaps with deoxynucleotides, and then finally there is strand sealing. The DNA ligase seals the gap between DNA fragments by forming phosphodiester bonds. Now, let us look into one important aspect of DNA molecule, which is the transcription process where DNA is copied into RNA molecules. So, we know about the primase copying the DNA into DNA primers or RNA primers, but in this case when we tell about transcription, it involves altogether different kind of process. Before understanding that, let us try to understand the genetic code, because the transcription has a purpose. The transcription won't stop at that stage it will further go on to translation, where the RNA molecule which has copied the information from the DNA will be further used for processing and making proteins. So, further understanding the concept of genetic code is very important, and in this aspect let us summarize the central dogma which is very important for the genetic code. So, as I have briefly mentioned about DNA getting copied to RNA and the RNA finally converting to proteins, the central dogma describes this flow of genetic information from DNA to RNA to proteins. Genes encode mRNA sequences which are translated into proteins, converting the 4 nucleotide genetic code into triplet codons that specify around 20 amino acids. This table describes the different codes, genetic codes against corresponding amino acids. For example, if we have 3 U's, first place U, second place U, third place U, this will code for phenylalanine. But if, for example, the third place is changed here, in this case A or G, it will represent leucine. But then in certain other cases, all the type of combinations available in the third place U, A, Z may represent a single type leucine. So, and then leucine again has 2 sets of say code, first and second having both UU, and in the second case the first place having all C. So, this table has been worked out for each and every amino acids, which are 20 amino acids. Now, there are certain codons, we call these triplet as codon. There are certain codons which do not code for amino acids, rather they give certain signal like stop signals. That is, once these signals are there, the transcription will be stopping over there, the process will stop over there. So, now mRNA translating will not proceed further once these stop codons are encountered. So, we will try to understand this process in more detail. So, some of the things that is important to know at this point of time is the degeneracy and termination, about which we have spoken in brief. Of these 64, 3 are representing the stop codon. So, 64 minus 3, 61 specify for amino acids. So, this 61 corresponds to 21 amino acids. So, many amino acids have more than 1 or 2 codons. So, this is what we call as genetic redundancy. So, this allows multiple codons to code for the same amino acid which we have described earlier. And then these 3 stop codons serve as termination signals recognized by release factors RF1 and RF2 in bacteria and ERF1 in eukaryotes that hydrolyze the peptidyl tRNA bond, ending the process of translation. The genetic code is nearly universal, facilitating comparison of protein coding sequences across organisms, but there is some kind of variations between the prokaryotes and the eukaryotes in terms of amino acids. The translation and the genetic code. So, let us look into this diagram first. This is an mRNA molecule having a directionality 5 prime to 3 prime. This mRNA is produced from DNA by the process of transcription. Once we have this mRNA in our hand, so you have U here in place of the T. Now, these triplets are represent one codon, and of course, depending on the reading frame, we may have many codons from a small stretch of mRNA molecule. So, translation will occur in ribosomes with transfer RNAs. This is a tRNA that recognize the 3 nucleotide codons on mRNA. Codons are read in the 5' to 3' direction in a non-overlapping and continuous manner. The reading frame is determined by the initiation codon, which sets the correct frame for transition. So, this is a large ribosomal unit and this is the small ribosomal unit, and then we have the tRNAs bringing in the amino acids, and there is these anticodon which will identify the triplet codons on the mRNA by complementarity. You can see U pairs with A C pairs with G over here. So, in this sequence 5' AC, GA, CGA, CG3, this can yield different peptide sequences depending on the start codon. Let us now look into the process of transcription. This is the process of the RNA synthesis from DNA molecules. The RNA sequence is complementary to the DNA template with adenine pairing with uracil instead of thymine. As already mentioned, there are 3 main types of RNA. Transfer RNA, these carries amino acids for protein synthesis. Then there are ribosomal RNA which forms part of ribosomes essential for assembling proteins. So, these are RNA ribosomal, this is the tRNA which is a type of RNA, and these ribosome structures, whether it is the large or the small subunit, has also rRNA. So, ribosomal RNA, and we studied about these in one of the earlier lectures. So, then there is a messenger RNA. These two do not translate and produce proteins, but the mRNA will be producing proteins. But these two are very much essential for the production of protein using messenger RNA. So, mRNA carries the genetic code for protein synthesis. In bacteria, genes for related functions are often grouped into operons and transcribed together from a single promoter, producing a polycystrionic mRNA with multiple coding regions separated by spaces. Each coding region is translated independently into different polypeptides. Archaeal mRNAs can also be polycystrionic. So, you can see here a coding strand, and this is the RNA polymerase which is producing the messenger RNA, thereby DNA is copied into mRNA by RNA polymerase. So, some of the important features of transcription is it requires a template strand, the DNA strand which is used to create complementary mRNA. Then there is a non-template strand or the coding strand, the DNA strand similar to the mRNA, not used as a template because the mRNA is complementary to the template strand and the non-template strand is complementary to the template strand. So, the non-template strand and the mRNA will be having a similar sequence with similar bases. So, another important thing is the +1 site, which is called as the initiation site, the location where the transcription begins. Then there is the upstream or downstream position. The nucleotides before the +1 site are upstream, negative numbers. After that are downstream, which are the positive numbers. Transcription requires unwinding of the DNA to form a transcription bubble where mRNA synthesis will be occurring. In prokaryotes like E. coli, a single RNA polymerase enzyme will transcribe all the genes. It consists of a core enzyme with 5 subunits like alpha, which has 2 units, then beta, and then there is beta prime, and omega and sigma factor, all in single copies. So, you can see the molecular size of these different subunits over here. Alpha is around 329 amino acids or 36.5 kilodalton. Beta is 150.4 kilodalton, beta prime is 155 kilodalton, and then omega is around 10.2 kilodalton, and these are the genes which produce these particular subunits of these enzymes. RPOA produces alpha, RPOB beta, RPOC beta prime, RPOZ the omega, and the functions of these subunits are: alpha helps in RNAP assembly, interactions with DNA and transcription factors for transcriptional regulation. Beta helps in DNA and RNA binding, RNA synthesis, NTP binding, RMP binding site, then also sigma factor binding. And beta prime helps in DNA binding, RNA synthesis, catalytic magnesium coordination, PP, GPP binding sites 1, and sigma factor binding, and omega helps in RNA folding, PP, GPP binding site 1. This is a structure. You have 2 alpha, 1 beta prime, and omega. So, this is known as the core RNA polymerase, which when couples to the sigma factor forms what we call as the holoenzyme. The sigma factor mediates recognition and binding of RNA polymerase to specific DNA sequences known as promoters. So, this will be binding to the promoters, and this will ensure transcription initiation at the correct and specific location. The complete RNA polymerase enzyme composed of the core enzyme and the sigma factor is known as the holoenzyme, and it is a kind of a sequential assembly. 2 alpha join to form alpha 2, to which a beta adds to form alpha 2 beta, then joined by beta prime, and finally omega alpha 2 beta beta prime omega, and finally the sigma factor, which recognizes promoters, will bind to it, and then it will form the holoenzyme, and this is the functional enzyme. Initiation of transcription in prokaryotes. So, how prokaryotic RNA polymerases starts the process. The sigma factor provides specificity by ensuring that RNA polymerases binds to the correct promoter sequence and initiating transcription at the proper site. Without the sigma factor, the core enzyme would not recognize promoter sequence accurately. So, holoenzyme minus sigma factor, the core enzyme, it is not able to bind to the promoter sequences, and that will lead to non-specific or inefficient transcription. Once transcription begins, the sigma factor can detach. So, sigma factor is like a person who goes and introduce you to the house you want to visit, and then after introducing you to the house you want to visit, it will go away. So, once the transcription begins, the sigma factor can detach, allowing the core enzyme to continue. The core enzyme has a crab claw or clamp jaw shape with an internal channel that runs the length of the enzyme. Eukaryotic and archaeal RNA polymerases have a similar co-structure, but include additional subunits. Let us now look into the prokaryotic promoters. What is a promoter? A promoter is a specific DNA sequence where the transcription machinery, including the RNA polymerase, attaches to initiate transcription. Promoters are typically located upstream of the genes they regulate. The sequence per promoter is crucial because it influences the frequency of transcription, determining whether it occurs constantly, intermittently, or rarely. So, here you can see a promoter sequence. It has 2 regions. the minus 35 region and the minus 10 region, and then this is the +1 transcription start site. And you can see here this RNA polymerase or the core enzyme, and this blue one is the sigma factor. This sigma factor recognizes this promoter, and after that it may detach after starting the process, and this RNA polymerase will do the transcription as discussed. Prokaryotic promoters share conserved elements across species, notably two consensus sequences located at -10 and -35 positions upstream of the transcription start site. -10 region, this is known as the Pribno box, and the consensus sequence is TAT. Then there is the -35 region, which the consensus sequence is TTGACA. The sigma factor of RNA polymerase initiates transcription by binding to the promoter. The -10 region, rich in ATPase, aids in DNA unwinding. During initiation, short abortive transcripts of about 10 nucleotides are produced and released early in the transcription process. So, this is not a single strike process. So, it may keep on trying several times. It is like kick-starting on a vehicle in old times, where you start the process again and again, and ultimately when the knock is optimum, the whole process begins. Then once the initiation is over, the next phase is the elongation phase. So, the mRNA synthesis begins after the sigma subunit is released from the RNA polymerase, allowing the core enzyme to synthesize mRNA in the 5' to 3' direction of about 40 nucleotides per second. And then the key steps in these elongosis process are: number one, DNA unwinding. The DNA double helix unwinds ahead of the polymerase to provide a template for RNA synthesis. DNA rewinding also happens. The DNA reforms its double helix behind the polymerase. So, then there is stabilization. Ongoing stabilization is required for the base pairing between the DNA and the RNA, and after the elongation process comes the termination. In prokaryotic transcription, termination occurs via two mechanisms. One is known as the row dependent mechanism, another is the row independent mechanism. The row protein catches up to RNA polymerase at a Z-rich region, triggering mRNA release. The row independent method, a CZ-rich sequence causes the mRNA to form a hairpin loop, stabilizing the polymerase. Weak UA interactions lead to the release of the polymerase and mRNA. Once termination occurs, transcription is complete. Now, let us study about another important process, the translation. Translation is basically the process of protein synthesis from mRNA. So, basically here the genetic information copied from DNA into mRNA is finally translated into polypeptides or proteins, and these are vital for cell function, as proteins are involved in nearly every cellular process, including replication, transcription and translation itself, not to speak about other metabolic pathways. So, overall they catalyze reactions and regulate gene expression. These complex and precise processes highly energy intensive. A lot of energy is required in the protein synthesis. Translation occurs in ribosomes, where tRNA delivers amino acids that are assembled into polypeptide chains based on the mRNA sequence. Ribosomes are crucial for efficiently assembling proteins, ensuring protein which are essential for cellular functions are synthesized accurately. So, once again let us look into the structure of ribosome, which has two subunits in prokaryotes. The 30th small subunit made up of around 16s ribosomal RNA molecules and 21 proteins. Then 15s large subunit contains 23s rRNA, a 5S rRNA and 34 proteins, and their function is to help in the protein synthesis. When the 30S and 50S subunit join together, they form the 70S ribosome. This ribosome moves along an mRNA strand to connect amino acids and build proteins in prokaryotic cells. The ribosome undergoes a cycle of association dissociation during translation. It binds to mRNA, synthesizes the protein, and then datassets to be used again. So, this we call as the ribosome cycle. Then there are tRNA binding sites. The ribosome has 3 sites for tRNA binding. First one is the A site, then the P site, and the E site. The A site is the amino acyl tRNA site, where the tRNA with the next amino acid binds. Then the P site is where the tRNA holds the growing polypeptide chain, and then there is the E site where the tRNA exits the ribosome after delivering its amino acid. Then let us look into the mechanism of protein synthesis in ribosome. Protein synthesis in ribosomes involves 3 main stages: the initiation stage, the elongation stage, and the termination stage. Initiation, the 30S subunit binds to the mRNA at the Sine-Delgarno sequence, positioning the start codon in the P site. So, you can see here the P site. The initiator tRNA carrying formyl methionine FMAT binds to this start codon, and the 50S subunit then joins the complex, forming the 70S ribosome. Now, next is the elongation, where amino acyl tRNA enters the ribosome's A site. You can see here the A site pairing their anticodon with mRNA codons. The ribosome forms peptide bonds between the amino acids in the A site and the growing polypeptide in the P site. The tRNAs then shift through the AP and E sites, with empty tRNA exiting at the E site. Then comes the termination step, when a stop codon reaches the A site. Here release factors will bind, causing the polypeptide chain to be released, and the ribosomal subunits to dissociate from the mRNA. Now, in this entire process, tRNA plays a crucial role apart from the ribosomal subunits. So, let us look into the structure of the tRNA. tRNA is transfer RNA, which is a small RNA molecule found in the cytosol, and various semi-autonomous organelles of all living cells. These tRNA molecules have a characteristic secondary leaf structure that resembles a clover leaf, featuring several key structural elements like there is a acceptor stem, and then T-C loop, and then there is a variable loop and anticodon loop and the D loop. So, this acceptor stem is a region where the amino acid attaches to the tRNA. It is formed by base pairing between the 5' and 3' ends of the tRNA molecule. So, this is actually a single molecule getting folded on its own, and this is the 5' and this is the T', and this is the acceptor stem where the amino acid will be getting loaded. So, extreme end of the 3' of the tRNA has a specific 5' CCA sequence 3', that is crucial for amino acid attachment. Then there is a pseudouridine loop named for its inclusion of pseudouridine, an unusual base. This loop is a distinct feature of tRNA. Then there is the D loop, which contains dihydrouridine, which is responsible for its name. This loop contributes to the overall tRNA structure. Then there is the anticodon loop. This loop contains the anticodon, a 3 nucleotide sequence that pairs with the complementary mRNA codon during translation. The anticodon is flanked by a purine base on its 3' end and a uracil on its 5' end. Then there is this variable loop which is positioned between the anticodon loop and the CU loop. The variable loop can range in sizes from 3 to 21 bases and varies among different tRNA molecules. After folding caused by intermolecular base pairing, a tRNA molecule has one end that contains the anticodon, which interacts with the mRNA codon and the CCA amino acid binding end. Attachment of amino acids to tRNA. How these amino acids get loaded into a tRNA molecule. What are the states of tRNA? Charged tRNA and uncharged tRNA. The charged tRNA has a covalently attached amino acid which is essential for carrying the amino acid to the ribosome during the protein synthesis. The process of attaching an amino acid to the tRNA is known as charging. Then there is uncharged tRNA. This tRNA lacks an amino acid, is considered empty until it undergoes the charging process. Amino acyl tRNA synthetases are enzymes. It comes in two classes based on the attachment sites. Class 1 is the enzyme that attaches amino acid to the 2' OH group of tRNA. They are typically single monomeric proteins. Then there is a class 2 enzyme that attaches amino acids to the 3' OH group of tRNA. They usually composed of 2 or 4 subunits, dimeric or tetrameric. Both classes are crucial for ensuring the accurate pairing of amino acids with their corresponding tRNA molecules, which is vital for correct protein synthesis during translation. tRNA charging process. The charging of tRNA is a crucial step in protein synthesis and involves two main stages facilitated by enzymes called amino acyl tRNA synthetases. Number one is the adenylation. The amino acid reacts with ATP forming an amino acyl AMP intermediate. This reaction transfers adenosine monophosphate to the amino acid, creating a high energy acyl AMP bond. Pyrophosphate is released in this reaction. Then tRNA charging. The amino acyl AMP intermediate binds to the amino acyl tRNA synthetase enzyme. The amino acid is then transferred from the enzyme to the 3' end of the tRNA molecule, specifically to the 2' or 3' hydroxyl group of the tRNA's acceptor stem. AMP is released during this transfer. The outcome of these steps is a charged tRNA with a specific amino acid attached, ready for participation in protein synthesis. So, this is in brief the diagrammatic representation of the loading of an tRNA molecule by the process that we have just described. So, how the translation initiation happens? Protein synthesis begins with the formation of an initiation complex. In E. coli, this complex involves several key components: the 30S ribosomal unit, subunit, the mRNA template, the messenger RNA that carries the genetic code. Then there are initiation factors called IF1, IF2 and IF3, which are involved in this process. Then there are initiator tRNA, a specific initiator tRNA called a tRNA FMET, which carries formyl methionine. It is usually removed after translation. In subsequent elongation phases, a regular methionine is inserted by tRNA. This is tRNA methionine and this is tRNA FMET. So, this needs to be remembered, which is a very, very important point. So, you can see here the tRNA with the first amino acid, and then there is this anticodon and there is the start codon. Then you have this large subunit and the small subunit, and this is overall the mRNA, and there is stop codon where the process will stop. Let us look into the process with a little bit of more details. It starts with mRNA recognition. The Del-Garneau sequence in E. coli mRNA located upstream of the start codon interacts with the 16S rRNA of the 30S ribosomal subunit. This interaction positions the ribosome correctly on the mRNA. Then there is the initiator tRNA binding. The initiator tRNA FMET binds to the start codon AUG on the mRNA and sometimes in the GUG, rarely. The methionine is formylated, forming a formyl methionine complex. IF2 helps facilitate these binding. Then there is a formation of the initiation complex. The 30th ribosomal subunit mRNA and initiator tRNA have met forms the initiation complex, and IF3 ensures that the mRNA is correctly bound to the 30S subunit. Then there is joining of the subunits. The 50S ribosomal subunit associates with the 30th subunit, forming the complete 70th ribosome ready for the elongation phase of the translation. And in this process, the energy source comes from guanosine triphosphate for the various steps in this initiation process, including the binding of the initiator tRNA and joining of the ribosomal subunits. Then translation elongation. Once the translation complex is established, the ribosome has 3 tRNA binding sites, A, P and E, amino acyl, peptidyl and exit respectively. In initiation, the initiating methionine tRNA occupies the P site, while the A and E sites are available for incoming tRNAs. Then there is a codon recognition and the ribosome moves along the mRNA. Each mRNA codon aligns with the appropriate charged tRNA anticodon. This precise pairing is crucial for accurate protein synthesis, as the ribosome would otherwise bind tRNAs randomly without mRNA. Then there is amino acid addition. Charged tRNAs will enter the A site where their amino acids are added to the growing polypeptide chain. The amino acid at the A site tRNA forms a peptide bond with the amino acids at the P site tRNA. This bond formation is catalyzed by peptidyl transferase, an RNA based enzyme in the 50S ribosomal subunit. The energy for peptidyl bond formation comes from the high energy bond between each amino acid and its tRNA. Then the next step is translocation. After peptide bond formation, the ribosome undergoes a conformational change, moving 3 bases along the mRNA. This shifts the tRNA from the A site to the P site, and the tRNA in the P site moves to the E site where it exits the ribosome. This movement is facilitated by elongation factors that hydrolyze GTP, providing the necessary energy for each step. Efficiency. In E. coli, the translation machinery can add an amino acid every 0.05 seconds, allowing for the synthesis of around 200 amino acid protein in about 10 seconds. Then finally comes the termination. Translation terminates when a stop codon is encountered in the A site of the ribosome. Here's how this process undergoes. Recognition is the first step. When a stop codon is positioned in the A site, it is recognized by protein release factors, which are analogous to tRNAs, but instead function to terminate translation. Then there is release factor action. These release factors promote the addition of a water molecule to the carbonyl end of the polypeptide chain attached to the tRNA in the P site. This reaction, catalyzed by peptidyl transferase, hydrolyzes the bond between the polypeptide and the tRNA, releasing the newly synthesized proteins. Then there is disassembly. After the polypeptide is released, the ribosomal subunits, both small and large, detach from the mRNA and each other. And then there is recycling. The ribosomal subunits are then available to be assembled into new translation initiation complexes. Meanwhile, the mRNA is degraded and its nucleotide are recycled for future transcription and translation processes. This process ensures the efficient release of the newly synthesized protein and the reusability of the ribosomal machinery and mRNA components. So overall, we can see here the schematics of this process where there is initiation. You have this tRNA with the first amino acid, which we already described, along with the large and small subunits and the mRNA, and there is a start codon and there is the stop codon, and then the initiation happens and followed by elongation and then finally termination. So with this we come to end of this lecture. Thank you for your patient hearing.